This is me Arzu here and I welcome you to my channel Mr. Teeth, where your dental concepts get dissected. So this is our part 4 video of the oral mucous membrane, and in this video we will be discussing with the cytokeratins, keratinocyte and the non-keratinocyte. Similarly we will be discussing with the taste buds. Okay, so today's video won't be much longer, and we will be discussing with those topics, and those topics are mostly asked in a short note, okay, in theory exams, and also some of the points, okay, that I will be discussing in the video itself. Then some of the points can be framed into the MCQ question, so you should concentrate properly. Okay, so now without delaying much, now let's start our today's video. So our first topic is the cytokeratins. Okay, so in cytokeratins, okay, so this is one of the favorite short note question, okay, mostly asked in the theory exams. So it forms the cytoskeleton of all the epithelial cell along with the microfilaments and the microfibrils. Okay, so cytokeratins are forming the cytoskeleton. Okay, so cytoskeleton, I think you have learned about this, okay, in cell chapter, cell biology chapter. So it forms the skeleton of the cell, means cytoskeleton of the all epithelial cell along with the microfilaments and the microfibrils. Okay, similarly after that it is present at the contact areas of the cell also. Okay, so it forms also the cytoskeleton along with the microfilaments and microfibril, and also it is present at the contact areas. Okay, so contact areas means like desmosomes, hemidesmosomes. Okay, so at those areas cytokeratins are present there. Okay, after that function as a stress bearing structure within the epithelial cell and are important in maintaining the cell shape. Okay, so maintaining the cell shape come under this cytoskeleton part, and this as they are the cytoskeleton. Okay, so just relate with our body. Okay, so we have bones. Okay, so that gives us proper shape of our body. Okay, so skeleton gives us proper shape, so that skeleton also help us in resisting the stress, okay, in resisting the forces. So it function as the stress bearing structure within the epithelial cell. Okay, so it is easy to understand. After that it is also called intermediate filament. Okay, so cytokeratins are also called intermediate filament because the diameter of those cytokeratins are in intermediate size. Okay, so the size of these cytokeratins is intermediate, which is between the larger and the smaller microtubules. Okay, so that is the case scenario here. So it is also called the intermediate filament. Okay, and this can be asked in MCQ question. Okay, so another name of cytokeratin is intermediate filaments, and there are 20 types of the cytokeratin present. Okay, and among that 20 types of cytokeratin, okay, we have classified those cytokeratins in two types. Okay, so type 1 and type 2, and type 1 is a basic and type 2 is acidic, and from 1 to 8. Okay, so cytokeratin types, okay, like collagen type 1, collagen type 2. Okay, so similarly the cytokeratin type 1 to cytokeratin type 8. Okay, so it comes under the basic category, type 1 category, and from 9 to 20 it comes under acidic and in the type 2 group, okay, of cytokeratin. After that they occur in the pairs of the combination of the type 1 and type 2. In this absence it will be susceptible to the proteases. Okay, so this is very important point you should remember. Okay, so as I have told here, there is a type 1 and there is a type 2. Okay, and always you should remember that. Okay, so there must be a combination, there must be a pair of the both the types, means one cytokeratin should be from the type 1 and one cytokeratin should be from type 2, means one should be basic and another should be acidic. Okay, so that combination of the cytokeratin makes it strong and it resists the proteases breakdown. Okay, so proteases means they are the enzyme which breaks the proteins. Okay, so this is susceptible, so this makes susceptible to this proteases breakdown. Okay, so this is very important to remember. After that, cytokeratin's profile reflects both cell type and the differentiation status in the different layer of the epithelial cells and of the different types. Okay, so this is very important. Okay, so cytokeratin profile. Okay, so amount of cytokeratin. Okay, so cytokeratin profile in both it reflects the both the cell type and the differentiation status in the different layer of the epithelial cell, okay, and of the different types. So this line wants to tell us that, okay, so cytokeratin profiles are those component, okay, so those are those things that tells us, okay, so what is the status of the differentiation of the cells in the particular layer or in a different layers of the epithelial cell, and also in a layer in a different types of the cell, okay. So the thing that is written here, okay, so you should also understand this. So cytokeratins have also its own importance. After that, for example, okay, so in suprabasal cells, okay, in suprabasal cell, in masticatory mucosa, okay, if you will see, we can see ck1 cytokeratin 1 and cytokeratin 10 is present there, and in the lining mucosa there is cytokeratin 4 and cytokeratin 13, okay. So this can be asked in MCQ exam, so you can remember this. And in the ventral surface of the tongue expresses the ck5, c6, ck6 and ck14, okay. Similarly, in the soft palate, ck7, 8 and 18 is present there, okay. So all those note I have taken from the Orbans, and this data is also taken from the Orbans, okay. So this can be asked in a MCQ exam, so you should remember this, I think. Okay, after this, now we will move to the our another topic, that is a keratinocyte, okay. So keratinocyte and the non-keratinocyte. So in the keratinocyte, so they are the epithelial or the epidermal cell, okay, that synthesizes the keratin and the cytokeratin, and also process the cytokeratin, okay. So cytokeratinocyte and the non-keratinocyte, okay. So I have told about this keratinocyte and the non-keratinocyte in the first video or second video, I think, okay. So there I have told that, okay, so either the epithelium is keratinized or it is a non-keratinized, but there is a presence of the both keratinocyte and the non-keratinocyte, okay. Many of the students have confusion that, okay, so in non-keratinized there is a non-keratinocyte, and in the keratinized there is keratinocyte, okay. So that is properly wrong, so that is not there. In Orbans you can see there, okay, so there is properly written, so keratinocyte or the non-keratinocyte, okay. So they both the cells, okay, are present in both the types of the epithelium, okay, but the quantity of the cell may differ from the one type of epithelium to the another type, okay. So same thing here also. So keratinocyte, so as it is a keratinocyte, it must contain the keratin, okay. So keratin is present there, and cytokeratin also present there, and also it possesses cytokeratin, as I have written here. So it synthesizes the keratin, cytokeratin also possesses the cytokeratin or the keratins, okay. So there is presence of the keratin and the cytokeratin in the cell, okay. So you can just take it simple. After that, keratinocyte, it increases in a volume in each successive layer from the basal to the superficial layer, okay. So with the increment in the layer, okay, so when we come from the base to the superficial layer, there is an increase in a volume of the keratinocyte, okay. So that is from this keratinocyte. And after that, this non-keratinocyte, so they do not possess the cytokeratin filament, okay, hence don't have ability to keratinize, okay. So as they are non-keratinocyte and they do not possess the cytokeratin filaments, okay, and they do not have ability to keratinize, so they are mostly present in the non-keratinized tissue, okay. So you can relate like that way, but both the types of cells are present in both the types of the epithelium. You shouldn't have to forget this line, okay. After that, mitotic activity or mesorative changes or desquamation is absent in the non-keratinocyte, but those all features are present in the keratinocyte, okay. So this can be asked in a viva, okay. Tell me the difference between the keratinocyte and the non-keratinocyte. So first difference you can tell this, okay, there is a cytokeratin filament in the keratinocyte, but this is absent in the non-keratinocyte. Similarly, the mitotic activity, mesorative changes, desquamation, okay. So about this desquamation also I have discussed earlier. So all those are absent in the non-keratinocyte, but they are present in the keratinocytes. After that, they are not arranged in a layers and do not form the desmosomal attachment with adjacent keratinocyte, okay. So as this keratinocyte, there is a cytokeratin, okay, and cytokeratin are present on the desmosomal area, okay. So that we can remember like that way, and that is the concept also, okay. So it is not arranged in a layer and do not forms the desmosomal attachment, okay, as they do not have cytokeratin, and also they are not in layers, okay. So this is also important to remember. and after that dendritic and appears unstained or clear in H&E stain. Okay, so they are mostly in dendritic appearance. Okay, so dendrite, I think you have studied in the neuron chapter. Okay, so neuron have a axon and the dendrons. Okay, so dendrite is there. Okay, so that type of appearance is present in this non-keratinocyte, and I will show you the picture. And also in this dendritic appearance, okay, we have melanocyte and the Langerhans cells like that of the dendritic appearance, but Merkel cells, okay, I will be discussing that they do not have the dendritic appearance, but commonly we can say that non-keratinocyte have the dendritic appearance and they appear clear in the H&E stain. Okay, so they do not be, so they are not stained by this H&E stain. For staining those non-keratinocyte, we require the other types of the stain. After that, identified by the special stains or the immunocytochemical method. Okay, so same thing I have told you. After that, they migrate to the oral epithelium from the neural crests or from the bone marrow, and this is very important to remember. Okay, so the origin of the non-keratinocyte, okay, it is taken from the neural crest cell. Okay, so neural crest cell means, I think you have studied this about the neural crest cell in a general embryology, and I will be not discussing properly here, so you should once study that properly to get proper concept of this neural crest cell. But in this chapter, okay, you just remember that non-keratinocyte is developing from the neural crest cell and it is migrating towards the oral epithelium. Okay, and that is very important line. This can be asked in a viva also and can also be framed into the MCQ question. After that, melanocytes. Okay, so about the melanocyte, so it is the first non-keratinocyte I will be discussing with. So in the first line, derived from the neural crest and the migrate to the epithelium in 11th week. Okay, so as I have told here, it is derived from the neural crest cell and they migrate to the epithelium. Okay, so this migration takes place in the 11th week. Okay, after that, present mostly in the basal layer of the epithelium. So in the basal region of the epithelium, so we have four different layers in a keratinized and three different layers in the non-keratinized. Okay, I think you have remembered that. So these melanocytes are present in the basal layer of the epithelium. After that, they secrete the melanin. Okay, so they secrete the melanin as they are the melanocyte, and they lack the desmosome but have the long dendritic process which connects with the other 30 to 40 keratinocyte. Okay, so they do not have desmosome as I have discussed here. Okay, so there is no cytokeratin, so there is no desmosomes. Okay, no desmosomal attachment, so melanocyte do not have desmosome but they have long dendritic processes. Okay, as I have discussed here also, so dendritic appearance is there. Okay, so there are dendritic processes there and which connects with the 30 to 40 keratinocytes. After that, melanin produced from it, okay, transferred through the dendritic process to the adjacent keratinocyte which stores pigment in the form of melanosomes. Okay, so through those dendritic process, okay, so the melanin that is secreted from the melanocyte, they are transferred to the other melanocyte. Okay, or transferred to the other keratinocyte adjacent through the dendritic process to the adjacent keratinocyte. Okay, so adjacent keratinocytes, so keratinocytes are those cells, so keratinocytes, okay, so they transfer those melanin through this dendritic process, and those are stored in a pigment form. Okay, so and that pigment name is melanosomes. Okay, so this is important and can be asked in MCQ exams. Okay, after that, oral pigmentation can be studied by use of the dopa reaction, silver staining technique, and the Mason-Fontana stain. Okay, so as I have written here about this non-keratinocyte, that okay, so they are unstained or the clearly or clear in H&E stain, means we require the important we require a special stain for the identification of the non-keratinocyte, and those specific stain for the melanocyte are the dopa reaction and silver staining technique and Mason-Fontana stain. Okay, so you can remember by your own way. Okay, so DSM or SDM you can remember by your own mnemonic, SDM. Okay, so you can remember like that way. Okay, so SDM, S means silver staining technique, D means dopa reaction, and M means Mason-Fontana stain. Okay, After that, in DOPA reaction, cells that contain the tyrosinase enzyme, they stain dark. So melanocyte contain tyrosinase and it is stained dark. Okay, so DOPA reaction. Okay, so the main concept of DOPA reaction is that, okay, so the cells, okay, that is containing the tyrosinase enzyme. Okay, so remember this enzyme, tyrosinase enzyme. So this tyrosinase enzyme is stained by in this DOPA reaction. Okay, and after staining they appear dark. So this melanocyte appears dark, okay, when they are stained with this DOPA reaction. So melanocytes secrete the melanin in underlying connective tissue when they are ingested by the macrophages positive for the DOPA reaction. Okay, so same thing is, okay, so same concept is there. Okay, so this is a proper mechanism how they are stained. Okay, and this is general overview. So the cell containing tyrosinase, they stain dark in a DOPA reaction, and how they are stained dark is that, okay, so melanin is first secreted in the connective tissue, okay, underlying connective tissue, and those connective tissue are ingested by the macrophages. Okay, and upon those macrophages the stain is applied. Okay, upon those macrophages takes the stain, okay, and they appear dark, so they are positive for the DOPA reaction. Okay, after that, number of the melanocytes do not determine the pigmentation. It is activity of the melanocyte that determine the pigmentation. Okay, and this is very, very important. Okay, so you may think that, okay, so if there is a lot of melanocyte present in the epithelium, then there would be a lot more pigmentation on the epithelium. Okay, but that is totally wrong. So number of melanocytes do not determine the pigmentation. Always you should remember. Okay, so number of melanocytes do not determine the pigmentation, but it is the activity of the melanocyte. Okay, so activity of the melanocyte that determine the pigmentation. Also the size of the melanocyte, number of the melanosomes. Okay, and melanosomes, as I have told here, so they are stored in a pigment form. Okay, so and those pigment are called melaноsomes, okay, which stores pigment in the form of melanosomes. Okay, so those are called melanosomes. So melanosomes in it and degradation are important. Okay, so activity, so the pigmentation depends upon the activity of melanocyte, okay, size of the melanocyte, and the number of melanosomes with its degradation. Okay, so with the degradation there will be releasing of the melanin, so that gives the dark pigmentation. So activity, size, number of the melanosome, not the number of the melanocyte. Okay, so you can get confused, I think. So number of melanosome, not the number of melanocyte. Okay, so activity, size, number of melanosome, and its degradation. Okay, so these are the four important points you should remember for the pigmentation. Okay, so in this Langerhans cells, okay, so they are the clear cells or the dendritic cells. Okay, clear cell means they are not stained by any stain, and dendritic cell, okay, and they are found in the upper layers of the skin and the mucosa epithelium. Okay, and I think you have remembered. Okay, so this okay, that I have told here, melanocyte. Okay, about the melanocyte, so they are found at the basal layer. Okay, so you should relate like this way. So as I have discussed with this melanocyte just now, so this is present in the basal layer, and these Langerhans cells are present in the upper layers of the skin and the mucosa epithelium. After that, they have convoluted nucleus and characteristic rod-like granules in the cytoplasm termed Bobbic granules. Okay, so convoluted nucleus is there. Okay, with the dendritic appearance. Dendritic appearance is of the cell, and there is a convoluted nucleus present in that dendritic cell with the rod-like granules present in the cytoplasm. Okay, termed as a Bobbic granules, and this is a favorite question of the by examiners. Okay, so they used to ask where is the Bobbic granules is present. So you should tell that in the Langerhans cell there is a Bobbic granules. Okay, after that it doesn't give the DOPA reaction. Okay, but it was given by the melanocyte. After that, stain positive with the gold chloride, ATPase, and the immunofluorescent markers. Okay, you should remember this. So melanocytes are stained by the DOPA reaction, Manson Fontana reaction, and the silver staining technique. Okay, so there were three different types of the staining technique to identify the melanocyte. And in this Langerhans cell, it is not stained by the Stopa reaction, but they are stained with the gold chloride, ATPase, and the immunofluorescent marker. And I will underline this, okay, and as this is very important. Okay, so stain positive with the gold chloride, ATPase, and the immunofluorescent markers. Okay, and I think you will remember this. Okay, and hematopoietic origin, so this is very important. Previously it was neural crest cell origin, and here is a hematopoietic origin of the Langerhans cell. And Vimentin type of intermediate filaments. Okay, so Vimentin type of intermediate filaments are present in this Langerhans cell. After that, it is involved in an immune response, and this is an important function of this Langerhans cell, and I think you have studied about this Langerhans cells, okay, in general pathology. So it is involved in an immune response. It migrates to the epithelium in a response to the chemotactic factors released by the keratinocyte to surface receptor of the Langerhans cell. Okay, so it just migrates to the epithelium in a response to the chemotactic factors. Okay, so chemotactic factors that are released by the keratinocyte. Okay, so keratinocyte are the cells that release the chemicals. Okay, so those chemotactic factors, those chemicals, okay, attract those Langerhans cells. Okay, may function as do the macrophage by picking up the antigen and presenting it to the lymphocyte either locally or at the lymph node. Okay, so this Langerhans cell, okay, I think you know this. Okay, so they are mostly the antigen presenting cell. Okay, and macrophage is also the antigen presenting cell. So what they do is, okay, so they just pick the antigen and they present it to the lymphocyte either locally, okay, so locally or at the lymph nodes. Okay, so that is the main function of the Langerhans cell. After that, they play important role in the contact hypersensitivity, anti-tumor immunity, and in the graft rejection. Okay, so they are mostly of the general pathology, but you can remember this. Okay, while you are asked to write the note in this Langerhans cell, so at that time you can mention all those points. After that, I will move to the another cell, that is Merkel cell, and it is present in the basal layer of the epithelium. Okay, so these Merkel cells are present in the basal layer, okay, like that of the melanocyte, but Langerhans cells were present at the upper layers of the skin and the mucosa epithelium. In second point, pressure sensitive receptor cell. Okay, so the Merkel cells are pressure sensitive receptor cells. Okay, after that, in the next point, commonly seen in the masticatory mucosa. Okay, so they are commonly seen in the masticatory mucosa, but usually absent in the lining mucosa. Okay, so this is also a very important line you can remember. Okay, so Merkel cells are commonly present in the masticatory mucosa. Okay, but usually absent in the lining mucosa. Okay, and in the next point, differ from other non-keratinocyte in that they are not dendritic. Okay, so this point I want to enforce much. Okay, so as I have told previously, in the general point of this non-keratinocyte, okay, so there was written that dendritic can appear clear and unstained. Okay, so here also in this Langerhans cells and in the melanocyte, okay, we have dendritic appearance, but in the Merkel cell we do not have the dendritic appearance. Okay, so in this point, as it is written here, ultrastructurally nucleus shows a deep invagination and the characteristic rodlet are present in the nucleus. Okay, and it was convoluted in the Langerhans cell. After that, electron dense granule is present in the contact with the axon terminal, and this is important. Okay, so electron dense granule, I will show you in the picture. So electron dense granule is present in this Merkel cell, that is present in the contact with the axon terminals. Okay, and in the next point, they are stained by the pus stain, and neural crest cell is the origin of this Merkel cell. Okay, so we have completed this about the Merkel cells. Now we will discuss with this lymphocyte and the polymorphonuclear leukocyte are also found at the various level of the epithelium. Okay, so so the fourth type of the non-keratinocyte that is present in the epithelium, okay, of the oral mucosa is that, okay, so there is also a lymphocyte, polymorphonuclear leukocytes. Okay, so we have neutrophil. Okay, about this polymorphonuclear leukocyte means neutrophils. Lymphocyte are present at the various level of the epithelium. Also, the keratinocyte can activate the lymphocyte through production of the interleukin 1, but can also inhibit it. Okay, so keratinocyte can activate or inhibit the production of this activation, okay, activation of this lymphocyte, okay, by the production of the interleukin 1. And I think it is easy to understand. After that, lymphocyte produce the gamma interferon that stimulate the keratinocyte to express HLA DRA antigen, okay. So lymphocyte produce the gamma interferon, okay. So they are, those chemical may be important, so gamma interferon, but it is not quite asked in exam, but you can just learn once. So gamma interferon is released from this lymphocyte that stimulate the keratinocytes, okay, to express the HLA DR antigen, okay. So we have discussed with the non-keratinocyte, and I think it is very easy to understand all those non-keratinocyte. Now I'll show you the picture. So in this first picture, as we can see here, this is a melanocyte, okay. So we can see the dendrites, and from the dendrite we can see that yellow color bowel like structure, okay. So that is a melanosome, and that contain the melanin pigment inside it, okay. And the dendrite are discharging those melanosome outside to the adjacent keratinocyte, okay. Similarly, after that we can see the another picture that is of Merkel cell, and in this Merkel cell we can see that there is no any kind of the dendritic processes here, okay. But we can see the electron dense granule, okay, which is of red colored dotted structure present there, okay, which is just adjacent to the axon terminal. So this is easy to visualize, okay. So Merkel cell, just you should remember that it is a non-keratinocyte but without a dendritic appearance, okay. And after that we can see the another cell, okay, which is again having the dendrite, and this cell is the as I have told, this is a Langerhans cells, okay. So this all the mechanism are shown here, okay. I have taken this picture from the Google. So this picture, so the mechanism is not our context now, so just you remember, just you visualize the image of the Langerhans cell, okay. So how they appear in the microscope. We have just got an overview, okay, how these cell appear in the microscope. Okay, so we have completed with the non-keratinocytes, okay. And now we will start with our last topic of today's video, that is a taste bud, okay. So as I have written here in the first point, so taste bud, okay, so they are a small ovoid structure, barrel shaped, okay, and intraepithelial organ, means it is within the epithelium, and it is about 80 micrometer high and 40 micrometer thick, okay. So I have drawn a schematic diagram here, okay. So this is a barrel shaped, and this is a taste bud, okay, and this is present within the epithelium. So this is a basal layer, this is a intermediate layer, and this is a superficial layer, okay. So this is a non-keratinized epithelium, just for now, okay. So this is a barrel cell that is within the epithelium, so you should remember like that way. And the dimension of the taste bud is 80 micrometer high and 40 micrometer thick, okay. In the next point, it extends from the basal lamina to the epithelial surface. So same thing is seen here, okay. So this is a basal lamina or basement membrane, and extend up to the surface of the epithelium. After that, in the next point, outer surface is almost covered by the few flat epithelial cells that surround the small opening, the taste pore that leads to the narrow space lined by the supporting cells, okay. So this outer surface, okay, it is almost covered by the few flat epithelial cell, okay. So I will show you the picture, okay. So this is a outer surface, okay, and this is a circular structure, quite a circular structure, okay. So it is covered by the flat epithelial cell, and that surround the small opening, okay. So at this, okay, I have made a quite larger here, but there is a small opening in the top, okay, and that is called the taste pore, and that leads to the narrow space lined by the supporting cells, okay. So when you move inside the taste pore, okay, so we can see the narrow space, okay, that is lined by the supporting cells are there, okay. So just you remember, it is a barrel shaped cell, okay, and there is a taste pore, and after you get inside the taste bud, from this taste pore we can see the space that is lined by the supporting cell. So you can remember like that way. After that, outer supporting cell arranged like the steps of the barrel, okay. So this is V, so they are like just steps, okay. So in the barrel we can see the steps are there, okay. So different steps is present there, okay. So how to explain, but I think you know this steps of barrel, okay. So like this structure are present there. I think when you will see the drum, okay, so in drum also there is steps are present there, okay. So their steps are called So those steps of the barrel, okay, are similar with that of the supporting cells, and inner and shorter ones are spindle shape, okay. So outer supporting cells are of steps of barrel, and inner and the shorter ones are like the spindle shaped. Okay, in the next point, now between the latter are arranged 10 to 12 neuroepithelial cells that the receptors of the taste stimuli. Okay, so between those cells, okay, the supporting cells, we can see the 10 to 12 neuroepithelial cell. Okay, and the name signifies, so this neuroepithelial cell means the epithelium also containing the axon terminals. Okay, so there is also the connection of the nerves. Okay, so that is the neuroepithelial cells. So those uh, act as a receptor. Okay, so those act as a receptor for the taste stimuli. So those neuroepithelial cell especially act as a receptor, okay, not those supporting cells. And after that, neuroepithelial cells are slender dark staining cell, carry finger-like processes at the superficial aspect and resembles hair like at the light microscopic level. Okay, so this is important. So they appear cylinder-like dark staining cells, okay, and carry the finger-like processes, okay, at the superficial aspect, and it resembles the hair like. Okay, after that, they are rich, okay, in plexus of the nerve found, rich plexus of the nerve found below the taste bud. Some fibers enter the epithelium and end in the contact with the sensory cell of the taste bud. Okay, so from this point you just remember, okay, so there is a rich plexus of the nerve, okay, is present below the taste bud. Okay, and those rich plexus of the nerve carry the stimuli, taste stimuli, okay, that is perceived by this neuroepithelial cell. Okay, so those are the particular receptors. Okay, those are the specific receptor for the taste, neuroepithelial cells. And in the classical view, okay, so taste are perceived in a different regions of the tongue and the palate. Okay, so in tip, okay, so if this is a tongue, okay, so if this is a tongue, so if this is a tongue, so at the tip portion we can perceive the sweet. Okay, in the lateral border, okay, so this is a lateral border, so there is a salty. And at the posterior side, okay, so in this posterior side we just receive this bitter and the sour. Okay, and also in this middle of this tongue. Okay, so this was the classical view. Okay, so this can be asked in a viva also. So sweet at the tip, salty at the lateral side, and the bitter and the sour at the palate, and also in the posterior part of the tongue, with the bitter at the middle of the tongue and the sour at the lateral of the tongue. Okay, so lateral of the tongue means the most lateral is salty, but between this bitter and between this salty, okay, there is a sour. Okay, so this region, okay, so this region there is a sour. Okay, so you just remember like this way. Okay, so this was the classical view. Okay, that in those region of the tongue we can perceive those type of the taste, but it is totally wrong, and it was told by the other authorities that, okay, so this type of theory is a classical and we shouldn't have to believe in this theory. Okay, but uh, the taste, okay, so that I have written here, the other authorities believe that the taste cannot be broken down. Okay, so it cannot be broken down into the four primary, sweet, salt, sour or bitter, but contains the range of the stimuli forming the spectrum, okay, of the sensation making of all the taste. Okay, so this is a modern concept about the taste. Okay, and about this picture I think you have seen in your school level books also. Okay, so in a sweet taste we just perceive from the tip, okay, salty from the lateral border. Okay, but that is a quite a classical view, but the modern views on this taste sensation is that, okay, so we get, so the modern view tells us that we just can't separate the taste sensation in primary four different tastes, okay, like sweet, salty, sour or bitter, but we can just say, okay, so those taste sensation form the spectrum of sensation, okay, from the range of the stimuli, okay, contain the range of stimuli. Okay, so those taste sensation contain the ranges of the stimuli, and those form the spectrum of the sensation making of all the taste. Okay, and after that, taste occur when a chemical substance contact a receptor cell in the taste bud. The reception fiber, uh, the reception fi- fires, okay, so sorry, reception fires the nerve fiber, so the taste may be a continuum or the composite of firing of the many fiber. Okay, so this line is supporting this theory. Okay, so this taste, okay, occur taste occur when the chemical substance, okay, they contacts a receptor cell. So receptor cell is the neuroepithelial cell, okay, and those reception by the neuroepithelial cell, they just fire the nerve fibers, okay. So they just fire the nerve fiber, and firing of those nerve fiber, they form the composite stimuli, okay, composite sensation, and those causes the taste sensation, okay. So that is a proper overview of this working physiology of this taste bud, okay. So now I will show you the picture. So this is a taste bud, okay, and in this taste bud we can see the gustatory hair present there, okay. Similarly we can see the taste bud at the above, okay, and we can see the neuroepithelial cell, okay. So that red outline cell, okay, with the red dots, okay, so they are neuroepithelial cell, and another white color background, okay, so black outline is there, okay, so that is the supporting cell. So just you should remember there are two types of cell, one is gustatory cell and supporting cell present in the taste bud, and the gustatory cell are responsible for the taste sensation, okay, and supporting cell just support those gustatory cell, okay. And another name of gustatory cell is a neuroepithelial cell, okay, so that I have mentioned previously. Now you are clear with this picture. So after discussing with all those topic, keratinocyte, non-keratinocyte, taste bud, cytokeratins, okay, and today's video was quite of theory based, okay, and they can be asked in theory exams, okay. So they are mostly asked in short notes of the theory exam, so you can prepare as such, okay. So now I will meet you in the next video. Till then, stay happy, stay healthy. So tata, bye bye, thank you.